Okay, so in the previous video I had talked about three things. Those are the nitrogenous bases, the pentose sugar, and the phosphate molecule. Molecule that make up your DNA. And so if you see over here in this DNA, you can see this much of that DNA, this part of the sugar phosphate backbone, and half of that latter is called a nucleotide, and it's essentially the building block of DNA. So this is another nucleotide, this is another nucleotide, and every single one of them are called nucleotides. So when you put all these three together, you get what you call a nucleotide. And what a nucleotide is a pentose sugar, which in this case is deoxyribose, and that's the fifth carbon. So we're going to number the carbons again. 1, 2, 3, 4, and 5. And on the first carbon you have that nitrogenous base, and this would be your deoxyribose. And on the fifth carbon you have that phosphate group attached. Now, since you know that the phosphate group looks a little something like this, it has say four oxygens, and so you know that one oxygen is attached to this end of the sugar molecule, and the other end is actually attached to the third carbon of this next molecule. So if you want to visualize it, so I've got some of these tiny nucleotides that I've made, and actually let me clear this paper up. So on a clear paper, nucleotides that I have made. So this is A, you have T, you have G, and C. So you know that A bonds with T and C bonds with G. So this phosphate over here would bond with the fifth carbon on this chain and the third carbon on the next chain. So this is 1, 2, 3, 4, and 5. So here on the fifth carbon you have a phosphate, and it's going to bond with the third carbon on the next chain, and that's exactly how it's going to look like. So you have A, T, G, and C, and you want to bond these two nucleotides to form the double-stranded structure. And what you're going to do is you're going to invert this so that it comes together in the nitrogenous base, because you clearly cannot attach it like this, right? So you have to invert it like this. So this is actually a C, so it comes with the guanine. So a cytosine goes with the guanine, and the thymine goes with the adenine. And so if you take a closer look at this thing, you can see the molecular structure. So here you have your pentose sugar, your nitrogenous base, here you have your pentose sugar, your nitrogenous base, and your phosphate molecule. So this entire thing would be a nucleotide. So that would be a nucleotide. And what happens is that you can see over here that the oxygen molecules on this side of the strand points upwards, and this side of the strand it points downwards. And so you kind of have like two parallel lines but going in different directions. And what we usually call parallel lines in different directions, we call them anti-parallel. So, and that's essentially what your DNA is. It's an anti-parallel chain. So you can see that the phosphate molecule is attached to the third carbon over here and the fifth carbon over here. So 1, 2, 3, 4, and 5. 1, 2, 3, 4, and 5. 1, 2, 3, 4, and 5. So when you look at the ladder, so imagine you're imagining this as a ladder, right? So when you look at it from the bottom to the top, so when you're looking at it as if you're going to go up the ladder, you can see that the phosphate, one end of the phosphate is attached to the fifth carbon first, and then the third carbon. So fifth carbon to third carbon. So it looks like it's going from the fifth carbon to the third carbon. So this strand is called the 5' to 3' strand of the DNA. And in this side, you could number it from here. So 1, 2, 3, 4, and I'm so sorry, 5 here. 1, 2, 3, 4, and 5. 1, 2, 3, 4, and 5 on this end. So it looks like this phosphate is attached to this third carbon first, and then the fifth carbon on the next one. Again, the third carbon here and the fifth carbon here. So this strand, when you look at it from the bottom to the top, you could see it going from the third carbon to the fifth carbon. And so we call this direction of this strand as the 3' to 5' direction, and this strand's direction as the 5' to 3' direction. And the main reason why we denote these strands by their direction is because we would be using these directions to find out which strand undergoes which type of replication when we study about replication of DNA in the future. Replication of DNA, and also transcription, and replication of DNA and transcription. It's very crucial that you know the directions of the strands, because there's going to be different types of replications that you're going to study later on. But for now, you should know that there are two types of directions. One is the 5' to 3' and the 3' to 5'. So next, when we come to the bonding of the nitrogenous bases, in the previous video I had told you that adenine bonds with thymine and guanine bonds with cytosine. And over here you could see that this is of adenine molecule. An adenine molecule, actually, to make this easier, let me just show you this picture over here. So I have two sets over here. You have adenine and thymine, guanine and cytosine. So this adenine molecule has bonded with the thymine molecule, and these dotted structures are hydrogen bonds. And that is because there's a hydrogen over here which is a little electron positive, and the oxygen is electron negative. And same way with nitrogen, which is electronegative, and hydrogen, which is electropositive. And over here you could see that adenine and thymine share two hydrogen bonds. And the best way to remember that is that AT, and T stands for 2, so AT2. And here in guanine you have 3 hydrogen bonds, so 1, 2, and 3, so 3 hydrogen bonds. Again with O and a H, H and N, O and H. So you should remember that adenine always bonds with thymine and guanine always bonds with cytosine. So the best way to remember that is I ate good chocolate. So IATGC, and that is how I remember it. And so when you come back to this structure, you can see that these two, because they have a double bond, you can say that this is adenine, and obviously this would be thymine, and this would be a guanine, and this would be a cytosine. So now you know every component of the whole DNA structure you can see that the whole nucleotide is present, the phosphodiester bond and its direction, and the way that the nucleotide, the nitrogenous bases bond with the other nitrogenous bases in the second strand, which is having the different direction, forms the entire DNA. So this is essentially your ladder. And this is how your DNA looks like, but in the future videos I'm going to be representing them by these more simpler formats which do not have all these molecules and atoms and everything, just for the sake of simplicity and an easier way to understand it. So I hope you understand this video, and thank you for watching.